Alright guys, we're going to continue our walk through the different classes of macromolecules that we had started in the last couple of videos. Last video we got through, proteins and carbohydrates, a lengthy talk on those two. Today will be a little bit shorter, but I want to discuss with you lipids and nucleic acids. And we're going to kind of cover these two topics in very similar fashion to what we did with the other ones. We're going to basically cover what the monomer structure is. We're going to talk about how we put them together into bigger molecules, and then we're going to talk about some of the, as we work our way through all this, we're going to start to discuss some of the different polymers that we see within each one and also their functions. So that stuff will be interspersed throughout all the material. But the basic plan is kind of like what we've gone through so far. Talk about the monomer, talk about how to put it together, and talk about some of the functions. So let's jump into lipids, okay? Lipids are commonly known as fats. The generalized structure, and you're going to see a much more complex structure, a much more detailed structure here in a second. But generally speaking, these things are greasy or oily nonpolar compounds. And the reason for that is because they have large hydrocarbon chains attached to them. And that's something you'll see in some of the diagrams. And so that's one of the things that also makes it nonpolar. Those hydrocarbon bonds, the carbon to hydrogen bonds, are highly nonpolar bonds. And when you have a bunch of them, you get a highly nonpolar molecule as a result. There are plenty of different functions for lipids here, all sorts of stuff. We see them just about everywhere in the body. One of the main ones is used for energy storage. It's what we commonly know we commonly know as fat, right? Adipose tissue is the stuff that our body stores lipids or energy as, and when we need it, our body will break it down and burn through that. Okay, membrane structure, another extremely important, and maybe even more important than your energy storage. But we see lipids used in the membrane structure. It's called the phospholipid bilayer, and we're going to spend an entire chapter talking about membrane structure and how it functions and things like that. So I want you to make sure you understand that when we look at the lipids, we see something that we're going to revisit a couple of times, so make sure you commit it to memory. Protection against desiccation or drying out. This one we kind of take for granted, but you guys feel the effects of this sometime for sure in winter. It's one of the reasons you ever have a baby brother or baby sister. You put baby oil on them because little kids have a tendency to dry out. They have very dry skin because they have a high surface area to volume ratio, which means the water is going to leave them and leave their skin a little bit quicker, a little bit easier. And so with babies, you coat their skin in this oil and it keeps the water inside. Okay, so it acts like a blocker for water. Sometimes that's a good thing, sometimes that's a bad thing. Okay, insulates against cold. We see this in many Arctic creatures, whales and penguins and walruses and big seals and sea lions, stuff like that, right? One we kind of take for granted but does play a role is the fact that it absorbs shock. The past 4 or 5 years I've done the cadaver dissection lab in Leroy, and some of you guys may have heard of that, and some of you guys may have even come to visit. But when we take the heart out of the body, one of the things that is really surprising is the fact that it's surrounded in almost kind of a layer of fat, and to really see the detail of the heart and the vessels and where the chambers would kind of be delineated, you actually have to scrape some of that fat off. And so that fat is there to help absorb shock to the heart. Okay, it's something you see with a lot of different organs. A lot of the organs have layers of, not big fat thick layers, but layers of lipids around them to help absorb any shock that might come about. Okay, and we also see lipids play a role in regulating cellular activities because they oftentimes manifest as hormones. Okay, and we'll talk briefly about how they manifest as hormones toward the end. So we won't spend a lot of time on the hormone one, but I wanted to at least mention it here as one of the functions. Okay, so let's get into discussing what is the monomer structure. We'll call it a single unit. It's a big single unit, which is why I kind of put it in air quotes. But the monomer structure of a lipid is called a fatty acid, and the fatty acid has long chains or a long chain of mostly carbon and hydrogen with a carboxylic acid group at one end, where we get that, that's where it derives its name from. This is a fatty, we call this a fatty or greasy, oily tail. With the acid at the end, the carboxylic acid at the end, we get fatty acid, okay? So when these guys are part of bigger lipids as a whole, they resemble long, flexible tails. And you don't have anything good right now. Okay, I'll use this iPhone charger cord. They're flexible like an iPhone charger cord or just a charging cord. Okay, for the most part, they're straight. Okay, they're linear, but they do have some flex or some bend. They can kind of wave around in this liquidy, aqueous environment that these things happen to find themselves in very often. Okay, so that is the structure of the fatty acid. That is one component of a larger lipid. So we're going to use multiples of these to put together to actually make some of the other structures that we're going to talk about here. Okay, so let's talk first about the different types of fatty acids. There are two different types of fatty acids. You've definitely heard of both of them. Okay, but we'll talk a little bit more detail about each type. Okay, unsaturated fatty acids are ones that you've heard of. I know you have. They are generally considered the good fatty acids. Because they tend to be liquid at room temperature, and so they're good because the bad ones tend to clog your arteries, and the reason that they clog your arteries is because they're solid at room temperature. Okay, so these guys, liquid at room temperature, they are characterized by one or more double bonds somewhere in that hydrocarbon tail. Okay, and those double bonds create little kinks in the tail. It almost makes it look like it's bent. Okay, and those double bonds again are important to the healthy nature of unsaturated fatty acids. It turns out that our body can break down these double bonds much easier than a bunch of single bonds. Okay, so for whatever reason, again, this is a Bergman-Christofferson question. Double bonds are more reactive. I know that much about double bonds, that they are more reactive, and as such, they will more readily break down in your body when needed. Okay, and most of these unsaturated fatty acids or fats come from plants. And this is why we say, oh, eating plants is healthy for you, right? Because the fat that they provide is not terrible. It's not going to kill you, usually, right? I mean, there are always exceptions to most rules, but yeah. Okay, saturated fats are a little bit different. They have only single carbon-to-carbon bonds in the fatty acid tails. So the big difference here is in structure, and it is this structure that creates the healthy versus unhealthy nature of the saturated fatty acids versus the unsaturated ones. In the saturated fatty acids, all of the bonds that connect the carbons are single bonds. Okay? And again, in unsaturated fatty acids, the carbons in that string oftentimes have at least one and maybe more than one double bond. Okay, so if I go back here to this picture, I would ask you in class, okay, what type of fatty acid is this picture showing? And you would say, you'd look at that and say, okay, there's all single bonds between all of the carbons. That's a saturated fatty acid. Okay, and the other way that you'll notice the difference is that the saturated fatty acids, the saturated fatty acids oftentimes are just stock straight. Okay? They're just absolutely straight like a pencil. Okay, whereas, as we mentioned before, the unsaturated fatty acids have a kink or a bend in them, and the reason for that is because of the nature of that double bond. It creates that shape, it creates that bent-looking shape in there. Okay, so that's one way to tell the difference between the two. These guys, the saturated ones, the way I tell the difference, because this is one of those things that, you know, it seems really easy at first. Okay, saturated is this, unsaturated is that. I always get them confused. Okay, I would always get them confused, and to help me remember them, I think of a sponge, right? A sponge with water. When you think of a sponge that is saturated with water, it is completely filled with molecules of water. Okay, so you put the sponge in there, you put it in and you squeeze it and you let it suck up as much water as possible. You pull it out. It is completely saturated with water molecules. You could think the exact same thing here about the saturated fats or the saturated fatty acids. those fatty acid tails are completely saturated with single bonds between the carbons. Okay? The minute you add one double bond that connects those carbons in one of those fatty acid tails, it becomes an unsaturated fatty acid. Okay? So that's always how I remember, that the saturated ones are completely saturated with single bonds between the carbon molecules. Okay? So again, these ones are considered bad because they are solid at room temperature. If you've ever seen a steak, right? A steak has fat on it. That gristle, that fatty, yellowish, whitish stuff on the steak, that is saturated fat. Okay, it is solid at room temperature. Okay, you have to heat it up before it starts to melt, and that's great. You do that, and when you cook out and you make stuff, you make meat, it tastes so good when you liquefy that fat and then you eat it, and then, oh man, lo and behold, once it's inside of you, it likes to harden back up again. Okay, and one of the things that it'll do, and why it's so dangerous, it likes to harden back up in your arteries and cause blockages. Okay, and those blockages cause all sorts of health issues, from possible strokes, high blood pressure, and obviously heart attacks is a big one. Okay, so most of these types of saturated fats come from animals, and we can literally see these things. Okay, oftentimes these guys are viewed as bad. All right, so let's do a quick question and answer. I want you to try to guess what kind this is. Now, this is a totally different view. This is called a space-filling model of a fatty acid. Okay, disregard these red molecules and look at everything below the red molecules. Okay, look at everything below the red molecules, and you tell me if that's a saturated or an unsaturated fatty acid. Okay, pause it. Take a guess. It is a saturated fat. Okay, it is a saturated fatty acid. And now, for comparison, we look at the other one. I bet you can guess which kind this is right off the bat. If you look at our definitions of saturated fatty acids versus unsaturated, this one should pop right out and say to you, this is an unsaturated fatty acid because it's got this kink or this bend in it. Okay, and now we see, okay, the actual difference, what that double bond is going to do. That double bond right here causes this part of the fatty acid tail to kind of jut out sideways. Okay, it causes that bend or the kink. Okay, there might be multiple double bonds, and if there was a fatty acid with another double bond in it, let's say right here, it's a good chance that it would bend the other way. Okay, whereas you take comparison this one, this one is about as straight as you can be, especially when you look at it compared to the unsaturated fatty acid. Okay, so that's a really, really easy, good way to tell the difference between them. Just look for that bend or that kink. If you're looking at a picture that doesn't have like a bend or a kink because of space reasons, that takes up more space to put a bend in that picture, always look for a double bond, because it is that double bond that creates that kink or that bend in the structure. Okay, all right. Let's now talk about how we put the polymers of lipids together. Just discuss the structure and the not necessarily the function, but where we can see these fatty acid monomers. We'll talk about how to put them together right now. Okay, when we put these monomers together into their polymer structure, they form something called a triglyceride. Okay, and again, this is probably, if you've gone to the doctor and had a blood test, this is one of the things that they will test for, your triglycerides. Okay, they'll look at your triglyceride level, and that will give them an indication as to whether or not you are in good, somewhat good health or not good health. And they'll say you got high triglycerides or low triglycerides, this is what you should do, or this is what you should do. Okay, this is one of the things that they look for. Now, the triglycerides are put together with the things that we see up here. You need a glycerol molecule, and you need 3 fatty acid tails. Okay, now it could be any number. It could be a saturated fatty acids or unsaturated. It doesn't really matter right now. You just need 3 fatty acid tails and a glycerol molecule. All right. Now here we got this is our third organic macromolecule. This is our third organic macromolecule, and it is our third type of bond, it is our third type of bond that describes the type of macromolecule that we're creating, okay? So, 3 ester linkages are formed between a hydroxyl group of the glycerol and a carboxyl group of the fatty acid. That will make more sense when you see the actual molecules in the next picture here, okay? But if you ever hear the term ester linkage, you should think to yourself, fat bond. Okay, so we've got sugar bond, protein bond, and now we've got fat bond. We've got glycosidic linkage, peptide bond, ester linkage. Okay, those are our 3 kind of official names for the different types of bonds that hold these monomers together in their polymer form. Okay, all right. So let's bring the picture up and see what we've got going on here. So I like this picture here, and I've included it because it shows, I hate that thing when it pops up here. It shows the glycerol molecule highlighted in purple. Okay, there it is, glycerol molecule highlighted in purple right here. And then it shows our fatty acid tails highlighted in yellow out here. Okay, let's just disregard this thing and act like this thing's not even here. Maybe you can't even see it. I have no clue. But anyway, the glycerol molecule, this purple thing with the 3 carbons, and then we've got it shows an O here, but that's because we've already got a reaction and a bond that has occurred. What normally happens on these glycerol molecules, that O is actually an OH. Okay, that is a carboxyl group. Okay. Oh, yeah, that is a carboxyl group. Okay, and the glycerol molecule has almost what looks like 3 docking bays. Okay, so like each one of those, look at that. Okay, so here's one docking bay, here's another docking bay, here's the bottom docking bay. And on each one of these, you will add a fatty acid tail. Okay, so there'd be a fatty acid tail added here, fatty acid tail added here, and then also one added here. So that glycerol molecule, which is this hand right here, is a way for you to add 3 fatty acids to make a much larger structure. Now, let's see if we can't figure out what type of reaction is needed to add the fatty acids to the glycerol molecule. I bet you can. I bet you can do it. I bet you can figure it out. That's right. It's what we've talked about already at least 3 times, 4 times up until this point. It is the dehydration, synthesis, or condensation reaction, whichever one you want to call it. Okay, and that's why I've included this little diagram. I couldn't really find, at the time I made this, I couldn't find a better diagram. But it shows what's happening, okay? This OH group, see that guy right there that's kind of highlighted in blue, that would actually be on the glycerol molecule. It would be right here. There would be an OH group here. Okay, it would be an O, this O that you see with the H next to it. And what we see here, this H would come from the fatty acid. Okay, and we see the H and the OH come together. And what happens when the H and OH come together? Water falls off. Water falls off. And then we get this ester linkage, this ester bond. Okay, and this happens 3 different times to add those fatty acids. So it's very similar to everything that we've talked about in terms of how these monomers are going to be put together into polymer structure. We're using that condensation reaction again. All right, let's keep on trucking here. Talk about a second type of polymer structure that is very important to our lives, in the lives especially of cells, are the phospholipids. Okay, this is another polymer-like structure that is put together by using the monomers. This time it's a little bit modified. The structure of the phospholipid requires a glycerol molecule, which we've already talked about in the previous one. It does basically the same thing. It acts as a docking bay. The phospholipid needs 2 fatty acids, and instead of a 3rd fatty acid, it needs a phosphate. Okay, so that's the big difference. Instead of 3 fatty acids here, we use a phosphate group. Okay, and that will make more sense as to why we need that phosphate group or what that phosphate group does when we start talking about cell membrane chemistry and cell membrane biology. Okay, So the function of the phospholipid is to maintain the membrane, the cell's membrane structural integrity. Okay, and for our cells, they are arranged in what's called a bilayer. All right, and I'll introduce that term here, but we're going to spend a lot more time on it when we get to chapter 7. Okay, so what you see in front of you is a picture of a phospholipid. Okay, we've got the space filling model, which I like to see. Just to kind of show you the kink in this one and how it correlates to what we see over here. And then we've got the structural model off to the left. Okay, I like the structural model because it shows where the glycerol is in relation to everything else that attaches to it. Okay, so we see these 3 CH2, this CH2, CH, CH2. That is the backbone of the glycerol molecule. Here is one docking bay for a fatty acid. Here is another docking bay for a fatty acid. These are the ester linkages we saw in the previous slide. And coming off the other docking bay, the third docking bay, is our phosphate group, which we see added right up here. Okay, that's the phosphate group. And so we put this together in a very similar fashion to what we'd put the triglycerides together, but it creates a much different or more unique type of molecule than what the triglyceride does. In fact, it has a completely different function. It's a very, very specialized function, in this case, as part of our membrane, our cell membranes. Okay, and so this molecule is a molecule known to be amphipathic. Okay, that is a term that will pop up again, I believe, in chapter 7. It'll kind of be on a slide, but I'm going to introduce it to you here. An amphipathic molecule is a molecule that has both a polar and a nonpolar end. Okay, so when we think about this molecule and what it's composed of, we see this phosphate group, which is positively charged. You barely see it, but there's a positive charge, there's a negative charge. This is charged. Okay, this has some kind of polarity to it. Okay, this has some kind of polarity up here. Whereas down here, the large hydrocarbon tails indicate to us that this is an extremely nonpolar part of this molecule. Okay, and that's going to play into how this thing creates the bilayer that we see in cells. Okay, now depending on how you introduce these things into different watery environments, they will form different types of structures. Oftentimes, if you just take a handful, let's say you had a handful of phospholipids, you throw them in some water, one of the things that will spontaneously be made is called a mycel. Okay, that's this thing here, where they will orient themselves in such a way that they that the polar head regions, the head part, the phosphate part, is hydrophilic. Okay, it's polar, it likes water. It will orient themselves to the outside, and it'll keep the hydrophobic, highly nonpolar tails isolated on the inside, kind of protected from the water. They don't like water, right? Nonpolar things don't like water. Polar things do like water. So this is what would spontaneously occur if we had thrown just some of those phospholipids into a cup of water. What we see in our cell membranes is something similar to that, but it allows for two watery environments. It allows for the outside watery environment as well as an inside watery environment that our cell really likes in terms of the cytosol. Okay. In fact, if you did watch, okay, so I linked a video for you guys with the amino acids and the proteins, talking about the origins of life and macromolecules and things like that. If you were to take the time and spend the 45 minutes and watch that whole entire video, they talk about mycels as possible early cell membranes of early life, okay? Because they are actually things that will spontaneously form when you throw some in some water. I think there's a segment where they actually show mycels being formed with the addition of just some random phospholipids into a watery environment. So maybe take the time and watch that. It's got a lot of really cool stuff in that video. Okay. All right, that'll do it for phospholipids and things like that. Let's talk briefly about waxes. I don't even know why I have this in there, other than to just indicate to you that a wax is a type of lipid. Okay, it's not going to be on the test, but I wanted to give you the kind of complete picture of all the different types of polymer structures that are created when lipids come together. Oftentimes these waxes are a lipid type coating for plants and sometimes animal parts. Okay, so other than that, not going to get into waxes. A little bit more time, I guess, on steroids. Steroids are a highly, oftentimes highly nonpolar. They're not fatty acid tails like we've seen. So they're a different kind of structure of the fat that we've not yet been exposed to. They are, in this case, ring structures. But again, they are highly based in carbon and hydrogen. Okay, the hydrocarbons. They're strongly based in hydrocarbon structure. Only in this case, the hydrocarbon structures have been twisted up and folded up into ring formations and connected together. Okay, so they're still highly nonpolar. They're still oily, fatty molecules because they are so highly nonpolar. But in this case, they just have kind of a different structure. Oftentimes, steroids that are used as components of animal cell membranes, and that's one of the things, again, in Chapter 7 that we'll see. We'll see some of these steroid molecules that kind of stick out of the cell membranes, and they act sometimes as communication, cell communication beacons, if you want to call them that. But these steroids can also be modified to form sex hormones. Okay, so we see sex hormones created from these steroids as well. All right. Let's jump into nucleic acids. Again, I'm going to approach the nucleic acids, this fourth type of macromolecule, the same way that I had touched on all of the other ones. We'll talk about some of the general stuff, some of the general structures and functions. We'll talk about the monomer structure and then get into the polymer structure on top of that. Okay. So when we discuss nucleic acids, there are two different types of nucleic acids. And when I talk about nucleic acids, I'm speaking of the nucleic acid being the big structure, the polymer structure. Okay? So when we discuss the fact that there are two different types of nucleic acids, the main ones that we're going to discuss are DNA and RNA. Okay? DNA and RNA. So when we discuss DNA, this is probably all just a crash course in remember things, so I should be able to skip through a lot of this here. It is double-stranded. It can self-replicate. It makes up the genes from which we get our proteins for, and it's passed from one generation to the next. Okay, RNA, in contrast, is single-stranded. Some of the components that make up RNA are a little bit different, and that's one of the things that we'll talk about here as we work our way through this part of the chapter. And it functions in actual synthesis of the proteins that are coded for by the DNA. Oftentimes, the RNA is like a throwaway copy, like a disposable copy of the DNA. So it is a very important part of what our cells use to help build our larger structure. And it's made from the DNA template molecule. All right. So this is kind of the flow of genetic information when we want to make proteins. I'm not going to spend a lot of time on this one, but I wanted you to see where it all kind of lies, and it shows us our two large macromolecules, our two large nucleic acids. We got DNA here, and we've got mRNA, which is a form of RNA, and it kind of shows you the role that each one of them has. DNA is our master copy of everything that all the instructions it takes to build. We want to keep that one pristine so we can keep building stuff if we need to. So we make a disposable copy in the mRNA. We send it out to the ribosomes, and then the ribosomes make tangible products from our actual instructions. Okay, so that's the flow of the information that these two nucleic acids provide. All right, let's discuss that monomer. Okay, the monomer of nucleic acids is called a nucleotide. Okay. The nucleotide has always got three things in common. Okay, three kind of generic things. Both the DNA and RNA are composed of nucleotide monomers that share some kind of structure. They have a 5-carbon sugar. Okay, this right here is the 5-carbon sugar, and the reason we know that is because it's in this ring structure that has carbons, hydrogens, and oxygens in a very particular ratio. Okay, that's the 5-carbon sugar in this case. It's made up of a phosphate group right here, a negatively charged phosphate group, and also one of 4 nitrogenous bases. All right. Now, that sugar that I just mentioned, the 5-carbon sugar, can be one of 2 forms. Okay, it can be a deoxyribose or it can be a ribose sugar. Now, there is only a very small difference between deoxyribose and ribose, and I'm going to show you that on this slide right here. If you look at these 2 pictures in the green down here, the one on the right is ribose and the one on the left is deoxyribose, and I'm going to leave the mouse still so we can see. If you look down at the very bottom of these molecules, at the very bottom there's 2 OHs at the very bottom of ribose, and then there's only 1 OH at the very bottom of deoxyribose. So if you compare the 2 there, they're basically the same, except for the fact that that deoxyribose is missing an oxygen that the ribose has. Okay, that's where it gets its name of deoxyribose. Deoxygenated. No oxygen has been put on that particular number 2 carbon. Okay, so that's the only real difference between ribose and deoxyribose, but just that little difference creates such a large change in the actual function of the molecule. It's just kind of hard to understand or put that into perspective. Okay, but those are your sugar molecules that you would create, or that are part of the nucleotide. Here we see again the phosphate group, and here we see the nitrogenous base. Now the nitrogenous bases could be one of 4. If we're talking about DNA, it could be one of 4. It could be either adenine, guanine, cytosine, or thymine. Okay? It could be any one of those 4 depending on the code that happens to be inside of your body. If we are talking about RNA, it could be one of 4 as well. It could be adenine, guanine, cytosine, or uracil. There is no T, there is no thymine in any of the ribose or RNA molecules. Okay, so keep that distinction apart, because we're going to revisit it again when we get to DNA and stuff like that next semester. Okay, and you'll obviously need it for this test as well. All right, so that's the monomer. Okay, there's our monomer. I just got done discussing here. There it is. Great. Let's talk about how we build the polymer. Okay, we're going to talk way more specifically about building the polymer of DNA when we get into chapter 16, I think. That's the one, 15 or 16. That's the one where we discuss DNA replication. And when we talk about DNA replication, it's an entire chapter discussing how DNA is built. Okay, so I'm just going to give you a very brief crash course here. Okay, and one of the first things that we have to understand here about the strands of DNA is that they have some directionality. Okay, and again, I'm only going to introduce it here. It's probably not going to be a heavy subject on this unit test, but it'll be a heavy subject on the unit that has DNA replication with it. Okay, now when we look at the nucleotides, let's just isolate in our vision this nucleotide up here. When we look at a DNA molecule, a strand of DNA, oftentimes the end of the DNA is labeled either 5' or 3'. Okay, and that indicates some directionality to that strand of DNA. And the way that the reason that this side up here is labeled the 5' end is because of how this ribose sugar is labeled. Okay, this is another crash course in organic chemistry. The way that they label the sugars, or the carbons in the sugars, they usually will start at the very top of the molecule. In this case, the top has an oxygen, so we don't start there, so we go next to the right. So this first one is called the 1' carbon. This little elbow here has a carbon there that they just don't write to save time. Then we go to the next elbow. That's the number 2 carbon. You see it's labeled as such. Then we go to the next elbow, the 3' carbon, and then the 4' carbon. And then, hey, we got this other carbon right up here, right above my pointer, that they call the 5' carbon. Okay? And so we know that this is the 5' end because that 5' carbon is the one that's exposed at the top. Okay, so that's why they label that side the way that they do. And if we look at the other end, and that thing covers it up again. I'm going to let it go away. But we look at the deoxyribose sugar at the bottom, we label it the exact same way, 1', 2', 3', 4', 5'. You'll notice that the one that's kind of isolated all at the bottom, the lower left corner, is the 3' carbon. That's why they call that end of the DNA the 3' end. Okay? So now that gives us some way of understanding how new nucleotide monomers are brought into and put together with existing nucleotides in a growing strand. The phosphate group of one nucleotide forms strong covalent bonds with the number 3 carbon of the sugar of the other nucleotide. So if we did this, we had let's say my pointer here is another nucleotide, comes flying in here, it's going to make a bond with this right here, as we see has been done right here. There's a bond between this phosphate group and that 3' carbon. There's a bond between this phosphate group and that 3' carbon. Okay, and so what we have at the bottom here is another exposed 3' end, and then another one when we add another nucleotide, and then another one, and so on and so forth. It just keeps building. And so you can only add new nucleotide monomers to this existing 3' exposed end. You can't add new ones to this 5' end. It's just not how it works. Okay, the bonds can be formed more readily down at this end. Okay, so that's how we build the polymer, and it gets really, really big, and it creates this this directionality within the DNA. And now we've got, let's say we put the double strand together, which is usually how we see DNA. Put the double strand together. This one runs 5' up here to 3' down here, and the other strand runs what's known to be anti-parallel. Okay, and these are all terms that are going to pop back into our lexicon next semester, where in this end is 5' and the other end is 3'. Okay, so that's what we mean by DNA's directionality. All right. So unlike some of the other monomer structures, nucleotides have more functionality than just being part of a larger polymer. Okay, yeah, I know that some of the sugars also had some functionality, like glucose and fruit, larger carbohydrates. Amino acids, as far as I know, don't necessarily have that functionality in their monomer structure. As far as I know, lipids don't have that functionality in their monomer structure, but nucleotides do. There are other jobs that nucleotides can do, and one specifically that is absolutely important to life on this planet. Okay, obviously we know that one of the big functions for our nucleotides is that they serve as monomers for nucleic acids. Okay, that's what we just talked about. We just showed you how you put a bunch of them together to build DNA, which is vitally important, right? One of the other big functions of nucleotides, the monomer structure of nucleic acids, is to transfer chemical energy from one molecule to another. And you ask, oh, wow, that seems kind of important. And it is, especially when you realize that it is called ATP, okay? And I want to just kind of point this out real quick here. If we look at ATP, that's the picture that you see at the bottom of this slide. That's a diagram of ATP. Disregard all the phosphates on the right side of the page, okay? And just look at that sugar molecule. That sugar molecule that you see at the bottom. That's a 5-carbon sugar, okay? Specifically, that is a ribose sugar. And then if we look at the molecule up to the upper left there, that's a molecule of adenine, okay? And that should sound familiar because it is one of the 5 nitrogenous bases that we can attach to the nucleotide, right? That's it right there. Okay. So that's adenine. That's one of the nitrogenous bases. Now, if we go to the other side, what do we know has a nucleotide structure? Has a sugar. It has a nitrogenous base. Okay, we've got the sugar, we've got the nitrogenous base, but it also has a phosphate group, which is what we see right here. Okay, it's got those 3 things. So we have a nucleotide right here. There's our nucleotide. Only thing that's changed is we've smashed these other 2 phosphate groups on the end of it. Okay, that's where we get the term ATP. ATP stands for adenosine triphosphate. Adenosine triphosphate. adenine is what gives us the term adenosine. That's why they call it adenosine, because it has adenine in it, okay? So when you hear ATP, think, oh, wow, all that is a heavily modified nucleotide that allows us to do things every day of our lives and be alive, right? So, nucleotides are vitally important. They make up our ATP. They make up our DNA, arguably two of the most important things that you can find in a living organism, okay? All right, so this is just kind of an overview of DNA in finality. I neglected to discuss the base pair matching, but I would hate for you to get to a test question on the Unit 1 test and be expected to know it and you weren't prepared for it for some reason. I think most of you guys know your base pairing rules, probably from 5th, 6th, 7th grade, something like that. But just brush up on them. When we put together this DNA double helix, we do so in a way that adenine always pairs with thymine and cytosine always pairs with guanine. Okay. When we discuss DNA, it's generally in a double helix form. It's double-stranded, and it's held together by hydrogen bonds. Okay. So I think that'll do it for our large macromolecules of life. Let me just double-check here. Yeah, that you can use that for review if you want. It's nothing I would worry about. But we are hereby done with Chapter 5 and also Unit 1 at this point. And I'm going to let you go at this point, and I will see you soon.